For over a century, the frameworks we use to measure animal intelligence have been limited by a specific historical lineage. We have evaluated cognition based almost entirely on what we can induce within the artificial bounds of a laboratory. The root of this methodology traces back to the early 1900s, when Ivan Pavlov discovered the principles governing how animals learn. He documented the mechanics of classical conditioning. When an animal learns to associate a neutral stimulus, like a bell, with a stimulus holding intrinsic meaning, like food, the association is driven by how closely in time the two are presented. Decades of subsequent research built a highly precise predictive model around this associative mechanism. Scientists can now predict how and under what circumstances almost any animal will form these specific responses. While accurate, this intense focus on conditioning created a scientific bias. It places an overwhelming emphasis on how researchers allow animals to learn, while often overlooking how they are biologically equipped to learn in their own environments. Modern behavioral research is shifting toward a different model, focusing on observational and social learning in mammals and birds. This research reveals that many species possess specialized cognitive frameworks. They do not merely associate stimuli through repetition. They extract data by watching members of their own group. This stands in contrast to automated conditioning. Social learning allows an animal to bypass a lengthy trial and error process and directly acquire the complex, multi-step sequences required for survival. The capacity for this type of learning is an adaptive trait. It is directly tied to a species' ecological demands and its social structure. And by isolating variables in a controlled setting, we can prove exactly how this mechanism functions. A field study in an Israeli pine forest presented researchers with a distinct ecological mystery. They discovered thousands of pine cones on the forest floor, stripped of their seeds down to the central core. The subject responsible for this methodical extraction was the black rat, Rattus rattus. Extracting these seeds requires a specific foraging technique. The rat must bite each individual scale off at its base, following the cone's natural spiral growth pattern from bottom to top. The researchers encountered an anomaly. Urban black rats completely lacked this skill. Furthermore, when these urban rats were housed with experienced cone strippers, they failed to acquire the technique. Passive proximity was insufficient. To determine if the skill was genetically inherited or socially learned, researchers designed a cross-fostering experiment. They swapped the infants of both groups to isolate maternal transmission as the primary variable. The first result clarified the mechanism. Infants born to urban mothers, but raised by cone-stripping foster mothers, successfully acquired the full stripping technique. The inverse held true as well. Infants born to stripping mothers, but fostered by urban mothers, failed entirely. The behavior is non-genetic. Acquiring this complex, sequential skill requires a specific, active maternal intervention. Simply being near an expert is not enough to bridge the gap. This raises a question of evolutionary drive. Why does the black rat expend the effort to learn this specific technique rather than foraging for more accessible food? Researchers measured the metabolic cost of the behavior by tracking the oxygen consumption of a rat stripping a cone inside a metabolic chamber. They then compared this cost to the raw caloric benefit of the pine seeds, measured by a calorimeter. The data revealed a significant disparity. The energetic cost of the stripping maneuver proved to be less than 10% of the caloric value gained from the cone, an acceptable profit margin. In the wild, naive adults can only learn the skill if they are provided with cones that have already been partially stripped. Juveniles, however, learn by stealing partially stripped cones from their mothers while they are feeding. Social learning functions as the biological mechanism that grants access to an exceptionally high-yield energy source, one that is otherwise metabolically inaccessible. To test the limits of this cognitive adaptation, we can look at a different model of social learning. In 1996, Bednikoff and Beta conducted a comparative experiment on seed caching behavior in birds. They selected two related species with different social structures. The Clark's nutcracker is a solitary specialist, while the Mexican jay forages in social groups. Their caching baselines reflect these differences. The solitary nutcracker hides up to 30,000 seeds across scattered locations for winter. The Mexican jay also caches food, but is significantly less dependent on these stores. The researchers designed a spatial experiment to test their memory. In a room featuring a grid of holes in the floor, a cacher bird hid food while an observer bird watched from a cage. 2 days later, both the cachers and the observers were tested for their discovery rates. The researchers measured their performance against an estimated random baseline. Look at the primary cacher results. Both the nutcracker and the jay performed significantly above random chance when covering the seeds they had hidden themselves. The critical variable was observational. Could the observers remember where they saw another bird hide a seed? Despite being a caching specialist, the solitary nutcracker failed. Its discovery rate as an observer plummeted to random chance. The social Mexican jay produced different results. These observers were just as successful at finding observed caches as the birds that originally hit them. Observational memory is not tied to general spatial intelligence. The nutcracker has a massive memory capacity, but its solitary lifestyle provided no need to track others. The Mexican jay's memory is a specific adaptation optimized for social foraging. Bringing these two models together provides a clear picture of how animal learning functions. The solitary nutcracker relies on an internal cognitive map, possessing no evolutionary equipment to exploit the work of others. In contrast, the black rat and the Mexican jay rely on extracting data from their social environments to survive. These capacities show that social learning exists as a specialized, species-specific tool, rather than a generalized trait that can be taught through mere proximity. When we evaluate animals through generalized conditioning models, we miss the actual architecture of their minds. We must measure their cognition based strictly on the specific ecological problems they are evolutionarily equipped to solve. Future behavioral research must move beyond the constraints of associative learning to truly map the cognitive architecture of the animal kingdom. We are grateful to the IELTS FOCUS for supporting this academic breakdown. True academic rigor requires constantly questioning theoretical intent and examining the strict experimental data.